So good afternoon, everyone. I'm Shovon Sarkar, a PhD student of Marduk University. So today I'm going to talk about predation of two resident ladybirds on invasive-based tomato potato psyllid. So before my talk, I would like to acknowledge the traditional owners of the land on which we are meeting. I pay my respects to their elders past and present and the Aboriginal elders of other communities who may be here today. So at first, I would like to talk about the tomato potato psyllids. It's how it's invasive in Australia, and their potential background, biological control in Australian field, the associated resident predator species which you can find in the field, and some findings of my result. So TPP is currently the biggest threat of Solanaceae crop and worldwide. So it's actually native to North America and Central America. And more recently, it was spread to New Zealand and Norfolk Island, which is a Western Territory of Australia. And in 2017, DPARD found TPP in Perth in Western Australia. So it is believed that TPP came to Western Australia from New Zealand by accidentally. So the TPP eggs looks like yellow in color and mainly deposited on the edge of the leaves or the abaxial surface of the leaf. And the nymphs are also green in color and which also found in the lower surface normally. And the adults are cicada-like structure, but smaller than cicada, of course. So there are many host plants which you can find in Australia. There are some plants called true host, which is on TPP can complete their whole life cycle and survive. So there are 20. The Solanaceae are the main true host for TPP. Sorry, Solanaceae are the main host for the TPP, and there are like potato, tomato, capsicum, which are the main host we can find in the Western Australian field. Other than Solanaceae, TPP can also survive on convolvulaceae and Lamiaceae crop. So TPP can damage directly and indirectly both. So directly, TPP can feed on tomato plant and which causes psyllid yellow diseases, causes the decline of chlorophyll from the leaf and which severely causes tuber development. And other than that, they produce some sugar-like structure, which causes many fungal diseases. And indirectly, TPP is a vector of CLSO diseases, which is Candidus librivector solanarium diseases, which causes zebra sheep diseases. And when we fry for chips, we can find the black color inside the potato tuber. So like, till now, CLSO was not detected in Western Australia. So there are more economic impacts for TPP invasion. For example, we need to do continuous monitoring to see if there is TPP or not in the field. And there is a great increased management cost for insecticide. Moreover, it's also impact on export-import business. For example, after invasion in 2017, the restriction and prohibition was imposed in all horticultural crops from WA to other states in case of export. So if we see some news article from after just invasion of TPP in Australia, we can see how it's how the farmer lost millions of dollars after invasion in Western Australia for only Solanaceae crop. So now the question is the management. So farmer still relies on chemical insecticide for management of TPP. However, some countries observed there are TPP already developed resistance against those insecticides. However, the Australian authority approved certain chemicals for temporary basis, but these chemicals are normally applied for other soft-brooding insects like aphid and other insects. So we have to think about the alternative. So in that case, I choose predators for alternative as biological control agents because predators, adults, and larvae can consume directly, and adult can fly longer distance if required, and the larvae are highly voracious, and they can cause more immediate control capacity than other natural enemies compared to parasitoid and others. So the main question is, what are the resident species we can find in the field, and what is the predatory potential in case of biological control? So to answer these questions, I did some field work in past 2 years, and the department actually established TPP quarantine area for TPP for the invasion of TPP. So yeah, even though it is a quarantine pest currently. So I did field survey in north region of Perth in capsicum tomato field, and in the south in this year I did field survey in the potato field. So I found these predators in the first year of field survey in different locations. So there are 4 types of ladybird beetles, some hemipteran insect, some merit bug, navid bug, and also there are high numbers of lacewing species we can find in the field. So these are the 4 ladybird species I found in the field, which are the major predators, that Coxella transversalis, Hippodromia variegata, as conformis, and Coxella sex muglagata. And I also found some green lacewing, also brown lacewing, some shieldback bug, and Nevis bug. So other than these predators, I also found some spiders in the field. So they are mainly attack spider. They like to directly attack insect pest compared to other spider who wants to build nest in the leaf. So from these 4 ladybirds, I choose here in ladybirds because they already observed preying in TPP in the field. They are available in commercial basis, and people can rear it easily as they are resident species. So they can easily adapt in the field and for adverse condition compared to lacewing and other species of predators. So in the field, I found higher number of Coxella transversalis and Hippodromia variegata, these 2, compared to other ladybirds. So that's why we choose this for the next experiment. So from the 2-sex live table study, I choose the age-specific survival rate and the reproduction. Here is the black line represents the survival rate, and the colored line is showing the reproduction of Ace variegata on TPP, and a common pest of TPP, common pest of ladybirds here is green pea aphid. So we can see that when I feed TPP on Ace variegata, the Ace variegata live longer compared to when I feed GPA. However, when I feed GPA, the Ace variegata lays more eggs compared to TPP, but in case of TPP, they're laying eggs in longer period of time. And we also found similar results for Coxella transversalis, that is another species of ladybirds. So that proves that both ladybirds can survive and reproduce on TPP compared to a common prey, green pea aphid. So in the next experiment, I supplied them different densities of TPP to test their predation capacity. So we found that both ladybirds show exhibit type 2 functional response here with a decreasing proportion of prey eaten with increasing densities of TPP nymphs until a plateau was reached, which is a proof that the potential predators for TPP in the field in the glasshouse experiment. So in the glasshouse, I did some case experiment, and here I supplied different densities of ladybirds in the case with tomato plants on 40 numbers of TPP provided in this plant. So we can see the population fluctuations and different treatments. So in different densities can effectively reduce TPP when we provide higher densities of ladybird beetles. So there are actually more than 40% for Coxella transversalis when we supplied 4 ladybirds per plant, and which is more than 36% and 48% adult when we supplied 8 and 16 ladybirds for Ace variegata. So there is a significant reduction compared to untreated control when we provide ladybirds in the plant. So this figure proves my findings. We see when I provide higher number of ladybirds in the plant, we can see the plant health is really better than when there is no TPP, there is no ladybirds in the plant. So, and to conclude, I can say that the Western Australia had resident ladybirds species for TPP, and which can be a potential biocontrol agent. And they already showed potential against TPP in the glasshouse and in the lab. But however, further field study is required and more study is required. For this, I'd like to go for field again this year and next year for more location in Western Australia. And I will do gut content analysis to confirm they actually eating TPP in the field, and also do DNA sequencing for predation confirmation. And I will also do prey preference and prey switching activity to know their response of predators to invasive TPP compared to other pests like green pea aphid and other aphids. So I'd like to thank for Agriculture Produce Commission, Potato Produce Commission for funding my projects, and from my collaborators from Biological Service, UWA, Harry Potter Institute, and TPERD for their supports. Thank you all.
Thank you, Shovon, and appreciate you finishing in a timely fashion. That leaves us with a couple of minutes for questions specifically to you before we move into more general discussion. So who'd like to kick things off with a question for Shovon? Just while people are perhaps collecting their thoughts, I'll take chairperson's prerogative and perhaps not so much ask a question, but reinforce, I think, to you the importance of the DNA-based gut content analysis work that you mentioned is ahead of you. I think that's going to be enormously important for developing your arguments, because, of course, showing that these lady beetles will consume the prey item of interest under non-choice lab conditions is only a small part of the jigsaw puzzle, and you've really got to show that under field conditions that is an important part of their diet. So I hope that goes smoothly for you. Okay, are there any questions specifically for Shovon? Let me just jump in the chat and see if there's anything. Here we are. There's one from Xu Wei asking, are there any reports for insecticide resistance in WA?
Not yet. Not yet. It's only observed in USA and Mexico. Even in New Zealand, there is currently no report of TPP, their pesticide. Okay.
All right. And I'll read these out, even though you can probably see them, Shovon. I'll read them out so people can see them, even if they don't have the chat box open. There's a question from Jackie Steele. Are the pesticides that are used to control TPP also toxic to the predators?
Yeah, actually, there is no currently used pesticide prescribed by the Pesticide Authority of Australia for TPP. So at present, they are providing some pesticide names which normally used for aphids and other insects, but I don't know there is effect on the predators. So in that case, I have to think about in future for conservation biocontrol and augmentative release, and how can we keep them stay in the field, like border crops or something like that.
Okay, thank you, Shovon. All right, we might just see if there's a final question specifically for you, and then we'll open things up widely for questions or comments more broadly. Is there a final question for Shovon about his very interesting talk? How far through your PhD are you, Shovon? Just a year or so?
1.5 years. I started in March 2020.
Good stuff. All right, Shovon, I can't see any more questions specifically for you, so we're going to open things up. Oh, sorry, I'm seeing a hand there from Jonathan Finch.
Hi there. Yeah, so I was just wondering when the psyllid becomes active in the year, and how does that marry up with the activity of the ladybird? Is there a bit of a lag, or do they kind of overlap in their activity, or do we know that at all?
Psyllid actually active on the summer season, and which is the way I found more ladybirds in that field. Actually, I have some data which is not in the slide. I put some yellow sticky trap to know their activity. When in that case I found more ladybirds and more predators like lacewing in the field when there are more psyllids. So there is overlap, right?
Thank you.
Thanks, Jonathan. Okay, well, look, let's open things up widely to questions or comments from anybody. Oh, Shovon, yeah.
Yeah.
You're
Thanks,
going to
Christian.
come back to that question in our chat
Yeah,
box.
I got the question. So that will be, I will use qPCR.
Just read the question out, would you please, Shovon? Because some people won't be viewing the chat.
What techniques you will use to analyze gut contents? I will use PCR to know the gut content analysis. qPCR. And for further, I will do DNA sequencing if there are some small, some light band show up for final confirmation.
Okay, good. All right then. So let's do what I said a short time ago. We'll open things up widely for questions.